Hello. If you want to produce a protein on large scale, so you need a vector to carry that particular gene so that it can be expressed by a cell, maybe a eukaryotic or a prokaryotic cell. So basically, vectors are cloning vehicles, or they are the carriers, and they could be plasmid from bacteria or bacteriophages, or yeast artificial chromosome or bacterial artificial chromosome, or it could be a plant or animal virus too. But how do we decide a vector? The choice of vector is decided by what is the insert size that you're going to use and whether the cell is eukaryotic cell or a prokaryotic cell. So suppose if you have this bacterial cell where you can see you have plasmids, so what property should be there in this vector which is going to carry your gene, the gene of the protein that you want to obtain on large scale? So if you see this particular plasmid, you will find certain properties are required. These are very important properties which should be there in the carrier. First one is having ORI, that is origin of replication, so that the vector is able to multiply on its own, so that the gene that you have inserted also multiplies. Second is selectable marker gene, presence of selectable marker gene. So selectable marker gene is basically the gene which helps you to identify the plasmid which is present in the bacteria. Okay, so those bacterial cells which have the insert, they can be identified by using selectable marker. So selectable marker can be antibiotic resistance, or it could be gene, or it could be laxi gene, or it could be green fluorescent protein. And then we also require a polylinker in the vector. So basically, what is a polylinker or multiple cloning sites? In this particular area, when you want to insert the desirable gene, then you require to cut it with the help of restriction enzymes. So the basic purpose of a polylinker is to have multiple cloning sites, so that you have more than one restriction sites and you have a choice of using many restriction enzymes. And then the fourth property is the size. The size of the vector should be small. Otherwise, what will happen? If you already have a big vector and then you put one more, insert the desirable gene, so the size will become bigger, and then you will not be able to put back into the bacteria. So just to recap, what are the four properties of vector? First is ORI, presence of selectable marker, which could be any of these three. Then third one is polylinker or MCS, and the fourth one is small size. All right, see you, bye.